Thank you very much. It's really a pleasure for me to have this opportunity. I grew up 10 miles from here in College Park. Coming back on a beautiful April week is not a hardship for me, certainly, and I'm happy to be able to tell you a little bit more about Zika virus. So we study a lot of emerging diseases in Galveston. Emerging diseases are defined as either newly discovered, diseases that are expanding their range sometimes, and probably many of these are familiar to you. If you look across just the bottom row there, you'll see Ebola, which last year was our major concern among these. You see Chikungunya, which 2 years ago was the major concern. So these things are just coming one after the other the past few years. Even one on the left there, you see anthrax that was in the US Capitol at one time in the past. So why do these diseases emerge and spread? There are a number of factors, but as I'm going to tell you today, one of the major ones is rapid air travel. An infected person can get on a plane, which we think happened in the South Pacific in 2013, arrive in the Americas with a virus and set it loose in a mosquito-borne transmission cycle at any time. We have expanding populations with very poor neighborhoods in the tropics, especially in Latin America, that are helping to fuel these outbreaks. We're changing our environment in many ways, introducing new mosquitoes from other parts of the world. And the viruses themselves have the ability to mutate and evolve very rapidly to take new opportunities that we create for them to circulate in new places and cause disease. So Zika is the virus this year that I think most of us are concerned about, but it's certainly not the last one to come. So let me tell you a little bit about the history of Zika virus. First of all, we don't know a lot about Zika virus. 2 years ago, virtually no one in the world was studying this virus, and we still don't know a lot about it, but one of the things we do know the most about is actually its origins in Africa. And it was first discovered in the Zika forest in Uganda, and in fact, one of the things we don't know about it is how to spell its name right, because you can see that the original Ugandan spelling had 2 I's, and some time along between now and 1947, one of those got dropped. But it was known fairly soon after that it causes a mild dengue or flu-like illness in most people, and that's still our understanding today. Most people don't get very sick from Zika virus. It's only a targeted group of people who are at high risk. So even 80% of cases are completely asymptomatic. People don't even know they're infected. They don't have any reason to be diagnosed. We've known for a long time that the virus is widespread in Africa and Asia, and many people are exposed to the virus, but rarely is disease ever reported. In fact, before 2007, there were only 14 cases ever described since 1947. And we've known for some time that there is at least some evidence that the virus can enter an urban transmission cycle where 2 different mosquitoes, Aedes aegypti and Aedes albopictus, can transmit the virus to people. So our understanding is that this virus originated in Africa in forest cycles that involve monkeys that are the reservoir or amplification hosts. And where I studied this virus in Senegal for many years, 3 different mosquitoes transmit. These are mosquitoes that live in forests. They don't have a lot of contact with people, so these viruses usually stay in this forest cycle and we never even know that they're there. But occasionally people become infected who enter the forest, or some of these mosquitoes can fly out of the forest to infect people. And then if those people arrive in a city where either of these 2 mosquitoes occur, Aedes aegypti or Aedes albopictus, they can initiate this human mosquito cycle. And then if they jump on an airplane, either if they don't become symptomatic or if they're before the symptomatic phase of the disease and they don't yet know they're infected, they can move the virus halfway around the world in less than a day. And if they're bitten by a mosquito in their destination, they can start this cycle in a new location. And that's what we've seen during the past couple of years. So this is just sort of a genealogy of the Flaviviruses, which is the family of viruses Zika belongs to. And Zika is a fairly close relative of a few viruses that you probably have heard of, including dengue viruses just to the left there. West Nile virus you can see about halfway down. And Japanese encephalitis and even yellow fever viruses that we've known about for a long time are relatives of Zika. So it's not completely surprising that Zika can cause a disease in people or even neurologic disease like West Nile and Japanese encephalitis do. But what's surprising is that Zika only seems to do it in fetus and infants that are infected during pregnancy. By the way, if you have any questions along the way, please feel free to ask and I'll take them. So the two mosquitoes that appear to be doing most of the transmission now are Aedes aegypti, which we think is the principal vector involved. And you know, if you wanted to design the perfect mosquito to transmit a virus that circulates from human to mosquito to human, you couldn't do very much better than Aedes aegypti because it has certain characteristics of its behavior and its ecology that are just about ideal. The only thing that's not ideal if you're a virus wanting to spread is that it's confined to tropical and subtropical locations. It can't survive in very cold winters like in the very northern part of the US or in Canada, for example. But it feeds almost exclusively on people. It tends to feed more than once during its reproductive cycle, which lasts about a week from the blood meal to develop the eggs to lay them to go back and look for another person to bite. And its larvae, the aquatic stage of mosquitoes is called the larva. They occur in water containers right around people's houses. If you have flower pots that have standing water, if you have an old tire in your backyard or some bottles or cans, even something as small as a bottle top can be enough water for Aedes aegypti larvae to go through their development to produce adult mosquitoes. And then after the larvae emerge to adult mosquitoes, the females, which are the ones that bite and transmit these kind of viruses, they like to enter your house and stay there. So they have this close association with people that's evolved over thousands of years. And they also feed during the daytime. And this is very different than the mosquitoes, for example, that transmit West Nile virus, which tend to feed at night. So it's very important that we're educating people that there are different kinds of mosquitoes with different periodicity of feeding, different behaviors, and we can't think of them all in the same way, and we can't control them in the same way. The other one is Aedes albopictus, which was introduced into the US only about 30 years ago. It's not quite as good of a vector as Aedes aegypti because it doesn't live in as close association with people, but it can occur further north. So if you go into the very far northeast of the US, you can find Aedes albopictus, but not Aedes aegypti. But we think that they're not as efficient, so the risk in those areas is probably a little bit lower. So this is the estimated distribution of these two mosquitoes, and this was just updated 2 weeks ago by the Centers for Disease Control. And you can see on top in the blue is where Aedes aegypti is thought to occur in the US. In reality, in many parts of the country, we don't know for sure if Aedes aegypti is there. Unless there's a very active mosquito control program or some kind of research program, nobody's looking for these mosquitoes. But you can see that this extends even north of here in Washington, D.C. In fact, this mosquito was discovered here in Washington for the first time just last year. It wasn't thought to occur this far north. And it may be that climate change is allowing Aedes aegypti to move further north over time as temperatures increase. And its eggs can survive winters a little bit further north because the winters are not as cold as in the past. This is another phylogenetic tree or genealogy for Zika virus. I'm not going to go into how we make these. We make them using computers and sequencing the genetic material of the virus. But I'm just going to show you what we conclude about the history of Zika virus. So it was first discovered in Uganda, where you see the yellow star there, but we don't know exactly where in Africa it originated, but we're pretty certain it did originate in Africa. We know a long time ago, at least decades ago, it spread from Africa into Asia. And in red are all the countries where there's evidence that the virus has circulated at some point in the past based on finding antibodies in people there. So if you're exposed to a virus, you respond by making antibodies, and they usually persist the rest of your life. So we can look at a sample of people to see who has antibodies to Zika virus, and that tells us that they've been infected sometime in their life. And so the virus, you can see, is all over sub-Saharan Africa, and then the orange locations are where the virus has been detected directly, either a person was diagnosed with an infection or a mosquito was collected that was infected, something like that. So it's probably very widespread in both Africa and Asia for a long time. But what started in 2007, when this virus was really considered unimportant and just really a curiosity, was there was an outbreak in Gabon in Africa, you can see in blue, and this outbreak involved urban transmission by Aedes albopictus there, and involved a few thousand people. The next outbreak that occurred was in Yap Island in 2007. Yap is in Micronesia. It's a very small island in the Pacific. Only about 7,000 people live there, but the CDC did a retrospective investigation and that they found that the vast majority of people had been infected on Yap Island during this outbreak. Then what really started to heat up was in 2013, the virus was introduced probably somewhere from Southeast Asia, but we can't pinpoint the location at this point, into French Polynesia. Now, French Polynesia, instead of having thousands of residents, has hundreds of thousands of residents. Once again, the majority of those people became infected. So now we had a large population of infected people, many of whom travel. Globally, they travel. So we had a lot of travelers getting onto airplanes not knowing they're infected with Zika. Remember, 80% of people never show signs or symptoms, traveling various places around the world, and they spread the virus to other parts of the South Pacific, but also to Brazil. And we think the virus arrived there sometime in late 2013. Mm-hmm.
So when you're seeing the spread from Africa to Asia before hitting the Americas, are you seeing cases of microcephaly?
Well, that's a great question, and I'm going to return to that. But the basic answer is, even if there were microcephaly in these areas, we probably would have never noticed it. And I'll explain a little bit more why in a moment. But the virus was probably undetected in Brazil for at least a year. And then in about a year ago, in northeastern Brazil, a major epidemic started where millions of people were becoming infected. And then travelers spread the virus into the Caribbean, into Central America, and other parts of South America. And we have now we're in the middle of a major epidemic all over the Americas and the Caribbean. And then also there's been an outbreak recently in the Cape Verde Islands, and we think that this virus reached those islands directly from Africa, from one of these monkey mosquito cycles that I showed you. So the current situation is there are 33 countries and territories with Zika virus epidemics going on, including Mexico. It's really impossible to know how many people have been infected by the virus, but in Brazil alone, the estimate is 1.5 million people. There have been 4,600 cases diagnosed in the whole region here. So why do we have millions of cases and only 4,600 of them have been confirmed through laboratory diagnostics? The reason is that most of these regions weren't prepared to do diagnostics, and as I'll explain a little bit more, this is a very hard virus to diagnose unless we catch people at the very earliest stage of their infection. 312 imported cases into the US so far, but I think that number is going to be growing as this outbreak heats up with the rainy season in the Caribbean and Central America. 6 cases of sexual transmission documented here in the US. This is the first of these mosquito-borne viruses that's ever been documented to be transmitted from men to women sexually. And we also know now that there are large amounts of the virus in both the saliva and the urine. So there may be other mechanisms of direct human-human transmission, but it's very hard to understand how much of this is occurring compared to mosquito-borne transmission. So as I said before, the vast majority of people develop a very mild illness or no illness at all. It's inconsequential to them. But unfortunately, there are 2 very serious diseases that we now have very strong evidence that Zika is causing. One is called microcephaly, where if the fetus is infected during pregnancy, and probably mostly during the first trimester of pregnancy, the virus reaches the brain and causes severe damage to the developing brain to the point where large portions of the brain don't develop fully at all, and these children are born with very small heads, and their microcephaly simply means a small head. But many of them have very severe lesions in their brain where the virus has done severe and extensive damage. Initially, it was hard to make the direct connection between these babies and the virus, but now we can do that in more than half of the cases in our study down in Brazil between our university, Yale, and the Fiocruz Institute. The other very serious outcome is called Guillain-Barré syndrome, and this can affect anybody in any age group. It's much less common than microcephaly. There have only been a few hundred cases identified, but this affects the peripheral nervous system rather than the brain. Our own immune response attacks our own nerves in the arms and the legs and the other parts of the body and can cause paralysis, which is usually only temporary, but it can be complete paralysis and it can occasionally be permanent. So this is also a very severe and debilitating disease. It takes years to recover from this disease typically. It can occur from other viral infections as well, but Zika seems to be a little better than most viruses at causing this. So what happened recently? Why all of a sudden do we have the Zika outbreak and the spread around the world? I think there are two main explanations that we really are working hard to test now in epidemiologic studies and laboratory studies. One is that the virus has changed. We know that these kind of viruses, RNA viruses, they mutate constantly. They can change very rapidly and change their ability to cause disease or to infect different animals or humans or mosquitoes. And it's possible that this happened with Zika. And one reason we're interested in this hypothesis is that Chikungunya virus, which was the last one to arrive in the US 2 years ago, we have very strong evidence that this virus adapted to use Aedes albopictus as a vector for the first time, and this allowed it to expand its geographic range quite a bit in Asia. And so we're doing experimental mosquito infections to look for evidence of this. But I think probably the more likely hypothesis, and this gets back to your question, Lynn, is that this virus in Africa and Asia is probably circulating in forests, maybe even in towns and cities in a more constant manner. And so people are exposed throughout their lifetime to this virus, like we are to many viruses like influenza virus or viruses that cause the common cold. And so they may be exposed little by little. Most of them are asymptomatic, they never know it. The people who are symptomatic don't have a disease that gets much attention unless it leads to Guillain-Barré or microcephaly, but these are very rare diseases. Pregnant women have about a 1 in 100 chance of their baby developing microcephaly. So if the infections occur little by little, rather than during a huge epidemic where millions of people are infected almost simultaneously, they're probably never noticed. And microcephaly can occur from other infectious diseases. It occurs at a fairly constant rate all over the world. So unless you see a sharp increase in the number of those cases, you will never notice a link with something like Zika. In fact, even in French Polynesia, where they had more than 100,000 people infected, they didn't notice microcephaly. It wasn't until the Brazilian experience, and they went back and looked at their records, that they noticed that there was a slight increase in the incidence, and they detected 8 cases during their outbreak. So I suspect that this is happening all the time, but only in very small numbers of people in Africa and Asia, and not getting noticed. And many women, by the time they reach childbearing age, may already be immune to the virus as well. But we're actively working to try to answer these two hypotheses right now. So one of the big challenges with Zika is diagnostics. And during the early phase of the infection, we have what's called viremia. That's when the virus is present in the bloodstream. And so we can take a blood sample from a person who has a rash or a flu-like illness, and we can detect the presence of the virus in their blood using a genetic test called PCR. And that's a very sensitive and very specific test that can be implemented in laboratories just with a moderate amount of resources and sophistication. The problem is you can see that about 4 days after the onset of the illness, the viremia disappears. Our antibody response eliminates the virus from our bloodstream. There's one other protein called NS1 that we may be able to detect. But basically, after the virus is gone, our antibodies appear in our blood. In response to the infection, we make antibodies, and that's what protects us from getting reinfected with all kinds of infectious diseases. The problem is that in places of the world where these closely related viruses like dengue circulate, if people have dengue antibodies, those dengue antibodies react the same way as Zika virus antibodies in this assay. So we can't tell if they've had Zika recently or dengue, or even if they've been vaccinated with yellow fever, that can cause a cross reaction. So in places like the US, if we have a traveler returning with infection, we're not exposed to dengue very often, very occasionally to West Nile, but most of us don't have Flavivirus antibodies. So we can diagnose travelers throughout this whole time period. But people in endemic areas, it's very difficult because most of them have dengue infection. So we don't know today, for example, in Brazil, how many people were infected last summer during the big epidemic, whether there are enough susceptible people that we're going to see another epidemic in the coming rainy season there. So the CDC has been updating periodically their recommendations for Zika, and the diagnostic test that's available is either through state health laboratories or the CDC themselves. Very few hospitals or private labs can do Zika testing. So they've limited the recommendations for testing to only those high-risk groups. So pregnant women who have traveled or could have been exposed to the virus are tested using the two methods I mentioned for the detection of the virus or the antibodies. Pregnant women who have had unprotected sex with a man who may have been infected with Zika during travels are also at high risk. Women who travel to an affected area, return to the US, and believe that they have Zika because they develop a rash or conjunctivitis or a flu-like illness are recommended right now to avoid pregnancy for at least 8 weeks when we're fairly confident that the virus is gone by that time point because our immune reaction clears it. And then the real question mark at this point is how long after infection can a man transmit the virus to a female partner? And we just don't know yet. So the CDC is recommending that men who may have been infected abstain or use protection for the duration of pregnancy of their partner. We'll probably learn more about that, and that recommendation may change. So there are a lot of challenges that we're facing with studying Zika and trying to control it. One is that I mentioned in dengue endemic areas, we often can't diagnose people. The other problem is that Guillain-Barré syndrome may be triggered by Zika virus in a way that a vaccine for Zika could trigger the same disease. And so we're going to have to be very careful in developing a vaccine for Zika. And third of all, the testing of these vaccines, and this is what we've seen with Chikungunya and then with Ebola, is that these diseases that only occur sporadically, we see a big outbreak and then the virus is suppressed to a lower level or disappears. It makes it very difficult for us to test new vaccines or drugs because we can't respond quickly enough to do the testing during the outbreak. And we have to develop ways where we can develop clinical trials that as soon as we see the start of an outbreak, if we test a vaccine when there are lots of people getting infected, we can determine whether it protects people or not. If we wait too long like we did for Ebola, then we don't have a good way to determine whether it protects people or not. So what's the current risk here in the US? I think that the best information we have comes from looking 2 years ago at Chikungunya, which has exactly the same transmission cycle between mosquitoes and people. And if you look back with Chikungunya, we had a lot of imported cases going on for many years, from 2006 to 2013. But once the virus reached the Caribbean and Latin America, we saw a big spike and we had almost 3,000 imported cases in 2014. And we're probably nearing that level of importation now for Zika. Although Zika, the number is only 312, remember that we're only seeing 1 out of every 5 cases that develop symptoms. The other people who are infected never know it, but they may be able to initiate mosquito transmission. So the other important point is that the first case in Florida 2 years ago was in early May. And I think that we're now reaching the point where we're seeing more rain in places like Florida, South Texas, even Arizona has had dengue outbreaks. That the onset of the rainy season, the increase in temperatures, the increase in imported cases, the risk is going to peak, I think, during the next few weeks. And then it's going to last through the early fall. And so we're going to have to do what we can during this time period to prevent the virus from getting loose here in the US, being transmitted by mosquitoes and infecting more people. So far, there's no evidence that that's happened. So what can we do to prevent this? Well, in the near term, our options are really only what we've learned in the past with diseases like dengue and chikungunya. We can use traditional mosquito control methods, but as I'll show you in a moment, we have to change the way that we do that when we've been focusing on West Nile for the last 17 years. We have to educate people to avoid contact with mosquitoes, especially pregnant women. But even a traveler who returns from, let's say, Central America or the Caribbean and never develops any symptoms of Zika could be infected. And they could initiate transmission probably for up to 2 weeks after they return. So we have to do our best to educate those people, even though they're feeling fine, especially if they're returning to the southern United States. They need to do everything they can to avoid being bitten by Aedes aegypti, because they could initiate the cycle that then would threaten everyone in the region. In the long term, I think the solution to Zika is a vaccine, but unfortunately, vaccine development does not occur quickly. Probably by the end of this year, we'll have some good, promising vaccines to start testing in animals, and maybe in the next year or two to start testing in people. But it's probably going to take at least 2 or 3 years to get a vaccine completely through the clinical trials to the point where the Food and Drug Administration could consider licensing it for use here in the US, or other governments around the world could consider that. So I think that unfortunately a vaccine is not going to be much of a solution for the current epidemic. We can also develop therapeutics like antiviral drugs. We have those for things like chickenpox virus and herpes viruses. But the problem with Zika is that the highest risk group are pregnant women, and treating pregnant women with any drug is a very delicate and risky matter. Some drugs that are perfectly safe for children and healthy adults can be very risky for the fetus. So we have to be very careful about how we go about that and do extra safety testing. And then I think we can develop some new ways to try to control Aedes aegypti and other mosquitoes. So why can't we just eradicate Aedes aegypti? And actually, this was almost done between the 1950s and 70s when yellow fever was causing epidemics all over Latin America. Many countries you can see on the left-hand side there in yellow got rid of Aedes aegypti altogether. The only ones that didn't, in fact, were a few in northern South America, some of the Caribbean islands, and the southeastern United States. But those efforts were ended in the 70s when DDT, which was the main insecticide used, which is extremely effective, was recognized to also be very toxic and environmentally harmful. So we don't want to use DDT anymore, and other insecticides we have are not as persistent, so they're not quite as useful. At that time, there were many authoritarian governments that would come into your house or on your property, make sure you had no standing water, impose very harsh penalties if you had Aedes aegypti in your backyard. Of course, in the US, we probably would never tolerate that as much as many of these countries that had authoritarian leadership at that time, and the politics have changed in Latin America. And then a lot of these cities where we find huge populations of Aedes aegypti have grown by orders of magnitude since the 1950s. So I think it's very unlikely that we can ever eradicate Aedes aegypti, but we can do various things to control them. Now, on the bottom here, you've probably seen this image in your own neighborhood many parts of the country many times. This is how we typically control mosquitoes here in the US. We spray insecticides in aerosols from trucks or airplanes, and the mosquitoes that we're usually trying to control usually stay outside. So these fogs of insecticide penetrate through the vegetation and they kill the mosquitoes. But the problem is Aedes aegypti isn't outside in the vegetation. It's right in your house, and those insecticides don't penetrate into houses. Even if they have open windows and doors, they don't penetrate very well into places like closets where these mosquitoes like to hide in a house. The larvae of Aedes aegypti are not concentrated in one location. They're scattered in small containers around your home. So the only way we can really do this kind of control is to go into people's houses or onto their properties to control these mosquitoes. And that's very expensive and labor intensive to go house by house to do this kind of control in a city of 20 million people like Rio or Sao Paulo. It's almost impossible. And also Aedes aegypti is developing resistance to many of the insecticides that we use for it. So our ability to do this is limited in the long run by that insecticide resistance. So there are some new methods being developed that are showing some promise. Some of them are very simple traps like this one that are designed to attract Aedes aegypti females when they're looking for a place with water to lay their eggs. These kind of traps, this one you can see it's made from a very simple 5-gallon paint pail that you can buy at Home Depot for about $3. Other very cheap plastic materials. And there's a sticky material on the inside that when a mosquito lands to lay its eggs, it gets stuck in the trap and dies there. And these kind of mosquitoes are showing a lot of promise. If you take one of these and you put 3 of them around each house in Puerto Rico, it's been shown you can reduce the Aedes aegypti population by more than half. So they're very inexpensive. If we can get people to adopt them and pay the small price, or the government can fund this, it can have an impact, I think, in the near future. In the long-term future, you've probably heard about some new methods that have been on the news a lot. One of them involves genetically modified mosquitoes. And this is a technology developed in the UK where a genetic element is introduced into Aedes aegypti. And when the mosquitoes reach the larval stage, it produces a toxic protein that kills the larvae. And these mosquitoes can be reared in a laboratory or a factory-like setting by just putting a very inexpensive antibiotic called tetracycline into the water, and that prevents this protein from killing the mosquitoes. So the idea is you set up a big laboratory factory-like somewhere in a city where you want to control Aedes aegypti, and this has been done in several locations. You rear millions of these mosquitoes in the laboratory. You can separate the females from the males when they reach the pupal stage because they're different sizes. And then you take the male mosquitoes and you release them out into the wild, into the city. Those male mosquitoes, their only purpose in life is to mate with a female. So they go out and look for female mosquitoes. They mate with them. Mosquitoes usually only mate one time in their life. So if a wild female mates with one of these transgenic mosquitoes, their offspring carry the gene, and when they reach the larval stage, they die. So essentially, these male mosquitoes sterilize the females in the population, and if you release enough of them, you can knock down the Aedes aegypti population by over 90% in some of these early trials. You rear them in the laboratory, but in the case of millions of mosquitoes, probably a room like this could support the development of that capacity. But remember, this has only been done on a very small scale so far. So the advantage is that the transgene, and everybody's worried about GMOs, the transgene in this case kills itself, so it doesn't persist in the environment. The male mosquitoes don't survive long. Their offspring die at the larval stage, and the gene is gone. So it's a very safe form of a transgene, and in my opinion, it's much safer than spraying toxic insecticides all over a large city, which affect many other insects that are beneficial, which can affect people if they're applied improperly. But the limitations are, this has only been done on a very small scale so far, in a very small city. And these mosquitoes, Aedes aegypti, don't fly very far. In their entire lifetime, they don't fly even 100 yards. So you can't just release the male mosquitoes in a couple different parts of the city. You have to release them everywhere. So how do you grow hundreds of millions of these mosquitoes, spread them out across a big city like Rio de Janeiro, and do this in a cost-effective manner? That's the real challenge, I think. And this may be problems that are overcome in the future, and this may be a viable way to control these diseases in 5 or 10 years, but not during the next couple of years. The other technology I'm not going to talk much about is a bacterium called Wolbachia that can be put into Aedes aegypti mosquitoes, and then it can spread through the population very slowly because of this very short flight range of the mosquitoes. But ultimately, these bacteria, they suppress the ability of the virus to infect the mosquito and to be transmitted. And this technology is being tested in several parts of the world and is looking very good, but again, it's only on a very small scale right now, a neighborhood within a large city of Brazil or Indonesia, for example. So again, this is a promising technology, but maybe 5 or 10 years from now, not in the next year or two. So I just want to close by trying to put Zika virus into perspective for you. So it's a terrible disease to get for a very small target population. That is pregnant women who are exposed mainly during the first trimester. And you've probably all seen the photographs on the internet of these babies. They're really heartbreaking. And I think that this is why there's so much emotional response and fear of Zika. But in the long run, Zika is not as important as many of these other mosquito-borne diseases I told you about, like dengue, chikungunya, yellow fever, even West Nile, which is still going strong here in the U.S. So I think for the next year, we're going to have the continuation of this major outbreak of Zika. If we have local circulation in the U.S., I think there's going to be a lot of panic people who have a pregnant relative or themselves are pregnant, and for good reason. If you know the virus is circulating mosquitoes somewhere near you, there's a lot of uncertainty. You can protect yourself, but not completely. But in the long run, these diseases are not going away, and I think they're going to be a bigger problem. So we can't continue this pattern of sort of jumping from emergency with one disease to emergency with the next, and also not supporting our biomedical research programs adequately in the long run. You know, the NIH, which funds most of the research on Zika, has had a 22% decrease in its budget in constant dollars since 2003. And it's not just the money that I think impacts our ability to get out ahead of these viruses, but especially our ability to attract the really talented young people to become scientists and work on these diseases. You know, the students who come to my lab, they find out very quickly that I spend huge amounts of my time writing grant proposals. Each one has about a 12% chance of getting funded. So it's really a very difficult and stressful lifestyle to be a scientist during this funding uncertainty, sequestrations, the budgets don't come out until well into the fiscal year. And I think it's really hurting our ability to get the best young people into science again. So I think in the long run, this is a problem we really need to solve. Just to close out, I want to do a little bit of shameless promotion for our campus and what we do. So as the congressman told you, we have probably the largest program in the world on these emerging diseases. We have some unique resources. The World Reference Center for Emerging Viruses and Arboviruses is the largest collection of these viruses. One of the reasons that we studied this virus Zika before anyone else is it had been in this collection since 1947. We have a vaccine development center that's one of only 5 in the world that's part of the WHO system for emerging diseases. We have the Galveston National Lab, the facility you see on the upper right. It's one of 2 high containment laboratories built at universities, funded mainly by the NIH. It's been running since 2008, and we can work with Ebola or Zika or Chikungunya or virtually any virus in this facility very safely at high containment. And we have the National Biocontainment Training Program, again, the largest program of its kind in the world. We train scientists literally from around the world to work safely on viruses like Zika. And you know, the best way to stop Zika is to detect it very early when it's still halfway around the world by training scientists there to do the diagnostics and do the research and try to control it before it starts spreading and reaches our shores and causes us major problems. So I think this kind of training is really critical to keeping these diseases from moving around the world the way that they are. We've been involved in a lot of the emergency meetings and organizing meetings with the NIH, the Pan American Health Organization, and the National Academy of Science. We published some of the first papers, including the first animal model for Zika, which is a special mouse strain that was developed that produces the same kind of disease that we see in babies and people that develop neurologic disease. So we think this is going to be critical for testing the first vaccines and drugs. And there are many scientists around the world who are working feverishly to come up with a Zika vaccine very quickly. But we have to have a model to test it before it can go into people. We sent a team of our scientists over the Christmas holidays to work with the Fiocruz Institute in Salvador, which is really the center of the epidemic there, to help them set up diagnostics and to do training We're developing new vaccines and drugs on our campus. The Brazilian government is actually funding UTMB to do some of this vaccine work to the tune of almost $2 million. And then we're developing these animal models so we can begin to understand how the virus is causing these neurologic diseases and how we can stop it from doing that. So that's all I have for you, but I'd be happy to take questions and stick around to talk if you have any particular concerns about Zika and in the areas that you represent of the U.S. So thank you for your attention.
Just as a clarification, that if a woman had Zika before she became pregnant, would she develop an immunity such that during her subsequent pregnancy she would not run a risk of microcephaly?
That's correct. I don't know if everyone heard the question. If a woman is infected with Zika before pregnancy, is she protected during pregnancy? And the answer is almost certainly yes. When we are infected with these viruses, we develop an immune response that probably protects us for the rest of our life. So the idea of a vaccine, it would be the first group of people we would want to vaccinate would be girls just before they reach childbearing age to protect that most vulnerable part of the population. But ultimately, if we can protect large numbers of people, even if they aren't in the high-risk group, if they can't contribute to the transmission cycle, we can suppress Zika and protect other people. That's the reason that we don't have usually measles in this country. Everybody doesn't get vaccinated, but so many people are immune that the virus can't spread person to person efficiently. Yes?
Would a ban on fetal tissue research hurt your ability to develop a Zika vaccine and also look to ways to treat infants when they're in your utero with the disease?
Almost certainly yes. The problem with Zika virus is if you take a normal mouse and infect it, basically nothing happens. And that's true for almost any rodent. We've tested all kinds of rodents at this point. Monkeys, like macaques, do develop viremia. We don't know yet if they develop microcephaly or Guillain-Barré syndrome, but a lot of the HIV research has the same problem. If you put HIV into a mouse, nothing happens. And some of the best work on HIV right now is being done by taking human fetal tissues, putting them into mice that can't reject those tissues, and now you have a mouse with the human cells and tissues that HIV can infect, and you can do research on how HIV replicates, causes diseases. And I think it's very likely, we call these humanized mice, that they'll play a major role in Zika virus research. So I think you're exactly right with your question there. Yes?
I have two questions. One is, did you say that the antibodies that you get that would be in your blood are very similar for those various, flaviviruses?
Right.
And if so, is it possible to have a vaccine against flavivirus that would just
Right.
get all of it? And then my other question is, you mentioned the great way to stop it is to go out to where it's going to start and teach people how to look for it. Are there NIH grants to do that kind of thing, or where would the money for doing that come from?
Yeah, great questions. So the first question about a vaccine that would protect against dengue and Zika and perhaps West Nile, there are efforts underway to develop exactly that kind of vaccine. There are some dengue vaccines that one just got licensed a few months ago for the first time in a few other countries, others that are in the late stage of clinical trials. And if those vaccines work well, the same way that they are made will probably work well for Zika. And the two can probably be combined to protect you from Zika, dengue, and maybe also chikungunya. There are also good chikungunya vaccines that have been developed. So I think that ultimately we probably can develop a multivalent vaccine to protect against all these diseases that occur in the same places through the same mosquito-borne transmission. The second question about funding for the places in the world where we think these outbreaks may come from. The NIH does support some overseas research. The work in Africa I mentioned at the very beginning to understand where these viruses originate, that was supported by an NIH grant that I had in the past to work in Senegal. We just learned a few days ago that the NIH is going to fund a project to work in Borneo in Malaysia, where we know Zika virus circulates, and that may be the part of the world where all of this got started. The U.S. Military also runs several overseas labs that do a lot of work on these diseases. In Bangkok, Thailand, the U.S. Army runs a laboratory that's made a lot of the major contributions to understanding dengue for many decades. And so the U.S. government does have ways to fund that kind of work, and there are international organizations that try to get scientists working together on these kind of projects. But I think Zika is going to help facilitate that even more in the future. Yes?
Is it fair to conclude, and I'm thinking I probably got my question answered a few slides back, that there are some concerns as to the effect of this organism on later episodes of pregnancy, like in the second or third trimester, because those studies are pretty new and ongoing?
So there are really only 2 good studies that have come out so far. The problem is when microcephaly was first recognized back around October and November in Brazil, the kind of studies that you really need to do to understand whether Zika is causing it and what the risk factors are, you have to do prospectively. You can get much better results and better conclusions if you do it prospectively. So there were some studies that just got started a few months ago that are now yielding results. And one of those is a study in Brazil where pregnant women at early stage of pregnancy were recruited into the study. They were followed through the course of their pregnancy. Some became infected, some did not. The ones that did not become infected with Zika developed no microcephaly. The ones that did, it appeared that the risk throughout pregnancy was not confined to the first trimester. There were bad outcomes even after the first trimester. Now, I mentioned in French Polynesia, they didn't notice microcephaly during the outbreak, but they did a retrospective study, and they found that the 8 cases that occurred there were all first trimester exposures. So it was a smaller group of women than the Brazilian study. So the evidence is a little bit mixed right now, but I think the safe thing to assume is that any pregnant woman could be at risk, but the risk may be, or earlier on, the risk may be higher. And again, the risk was quantified in this Polynesian study to be about 1 in 100. So it's not a high risk for each pregnant woman, but when millions of people are getting affected in a place like Brazil, then you start seeing thousands of cases, and I think that's where we are now. Yes?
Is there any way in the early stages of developing a vaccine to see if it will result in, or could result in GBS?
This is going to be a big scientific challenge. So the first thing we need to do is understand what triggers GBS with Zika virus, and we need to develop an animal model where we can infect the animal, GBS develops, and we can look to understand what's triggering that. But what we know, the concern at this point is that when we look at the sequence of the genetic material from Zika virus and we compare it to our own genes, there are some proteins that have portions that are very similar between Zika virus and our own proteins. And the fear is that when our immune system develops when we're very young, we learn to recognize what's ourselves and what's different. And that's because if our immune system was constantly attacking our own body, we couldn't survive, but we have to be able to attack something new coming in, like a bacterium or a virus. So it may be that if Zika virus infects us, and it has some proteins that are very similar to our own, our memory to remember what's ourself sort of gets forgotten for a while, and we develop an immune response to Zika that also attacks our neurons. And if that's the case, and we don't know for sure, that's just based on comparing the sequences, but if that turns out to be correct, then we have to make sure that those proteins are not included in a vaccine. And we can probably do that, but we need to understand it very well before we would ever want to test a vaccine in humans. Yes?
What is the background rate for microcephaly without Zika?
Yeah. So in Brazil, for example, in the entire country, they typically see 100 to 200 cases per year. and I think Brazil has a population of about 200 million. That number jumped to somewhere between 4,000 and 6,000 cases in the last year. And that's really the jump in microcephaly and the cases being in the same place where Zika was circulating. That's what led to the initial suspicion, very strong suspicion, that Zika was causing this. And now we have very direct evidence for that as well.
So factor of 10 to 20.
Yeah, exactly. And Guillain-Barré syndrome also has gone up by a factor of about 10 to 20, but the incidence overall is much lower. Yes?
So we talk a lot about Brazil. With them having the upcoming Olympics, so there's going to be fans and athletes from all over the world in the U.S. going down there. What can be done to maybe reduce the spread of all that?
Yeah. Well, the Brazilian government, I think, is doing pretty much everything they can. They're literally mobilizing the military to do mosquito control. Again, to put this in perspective, the athletes going down, unless a pregnant woman or the partner of a pregnant woman is there during the Olympics, or the fans going down, the other people are not at risk. If an athlete gets infected by a Zika virus, a distance runner or a wrestler, or any kind of sport, they may get a little bit sick and may not be able to compete effectively, but it's not going to have a long-term consequence for their health. It's only that high-risk group. So unfortunately, I think athletes are already starting to pull out of the Olympics, probably because they're seeing Zika on the news so much and they're not reading enough to understand who is really at risk and who's not, that they're exaggerating their own risk. So I think that if everyone was well educated, the Olympics could still take place with not a lot of disruption. But unfortunately, I think that unless people really get well educated, they panic when they see these images in the news. They don't understand the true risk, and so the Olympics may be severely affected. I don't know what's going to happen. And unfortunately, Brazil has a major economic problem, a major political problem. You throw Zika on top of that, and it's really a terrible, kind of a perfect storm for Brazil right now. Yes?
I know the media loves to scare us. And the other day, I heard just like a one-line thing on the radio, Zika now presenting more terrible possibilities or something like beyond microcephaly.
Right.
And then could you please translate this?
Oh, okay. So on the left, it says, just a little clean water, please. And that's a mosquito carrying Zika, Chikungunya, and dengue, this triad that we've seen. And on the right, the mosquito on the left says, are you carrying Chikungunya? And this is a pre-Zika cartoon. Or today it would probably say, are you carrying Zika? And the one on the right says, no, I don't like to be fashionable. It's only carrying dengue. But back to your question, I think what's triggered some of these recent news stories is that the WHO and then the CDC put out news releases that the evidence, in their opinion, is finally so strong that there's no doubt that Zika virus is causing these bad neurologic diseases. I don't think that there was any dramatic new scientific finding that triggered that. It was just the accumulation of evidence, and they finally decided. And I think they're conservative organizations. So I don't think anything dramatic has changed our thinking about Zika in the last few weeks. But those announcements got a lot of attention by the press. I was getting a lot of requests to talk to the press about what this really means. It really just means that the accumulated evidence has finally reached a point where they thought that they had to put that out. I think that the other thing that we don't know about Zika is we can diagnose these microcephaly cases in late pregnancy through ultrasound or after childbirth, and it's very clear from the damage to the brain that a lot of these children are going to have severe disabilities the rest of their life. What we don't know yet is whether there are milder cases of infection in a fetus that don't produce a small head or lesions we can see in the brain through imaging, but may have an effect on learning or motor skills that don't appear until years later during development. And I think that's really the next stage of what we may not learn for a couple years about if we're only seeing the most severe cases now and just the tip of the iceberg. Yes.
We're getting actual effects of Zika during the Olympics while people are there. When they go back to their countries, do you think it would promote the spread of it elsewhere?
It could, and I would certainly hope that the Brazilian government, the Olympic authorities would do their best to educate everyone traveling to Brazil, like I think our CDC is trying to do and our state governments, that even if you go to the Olympics, you have a great time, you feel fine, you fly back to South Florida, you can infect mosquitoes and start the cycle without knowing it. So it just takes a couple of weeks of vigilance after returning home to reduce that risk a lot. And I hope that that word will get out in Brazil. Yes?
This is more of a general question. So in the long run, in like the U.S., as there's climate change and these mosquitoes are moving further north, do you see there being an increased prevalence of these tropical diseases within the U.S. and how do we combat that?
Well, I think it's almost certain that the distribution of Aedes aegypti will continue moving north, and in South America south and places like China, we'll see new regions at risk. We can't predict entirely what will happen because climate change has a lot of different effects, and these mosquito-borne viruses they have complicated cycles. So, for example, if a region of the world gets warmer in the summer, the mosquitoes may follow the increasing temperature, but if people stay inside their houses more, here in the U.S. where we have air conditioning, they may be exposed less to some of these viruses. So it's complicated. It's not so simple that temperature increases, the mosquitoes come, then the virus. But certainly in some parts of the world where you can't afford air conditioning, probably they're going to see the increased risk from these mosquito-borne diseases.
So these mosquitoes can't live or survive in an air-conditioned house because you say they like to live in homes, so they couldn't survive.
Well, they can if you let them in. I think that in a house in South Texas or South Florida, if you leave the door open for 5 minutes while you're unloading your groceries or whatever, some mosquitoes will certainly come in. But if you compare that to a typical house, I was in Salvador, Brazil, about a month ago in the wealthiest neighborhood of the city. These were homes that probably in the U.S. would be at least $1 million, in Florida or in Texas. And even these wealthy people had their doors and their windows wide open all day long. So it's just a different culture. It doesn't kill them directly. It just the fact that your doors and windows are closed is the main advantage of preventing their entry, I think. Yes? Yeah, that's another question that comes up a lot and is a good one. Probably if we eradicated many different mosquitoes, we would have environmental effects that would be difficult to predict because mosquitoes are food for a lot of animals like bats and other insects. But Aedes aegypti is different because Aedes aegypti, it's a mosquito that originated in Africa, just like Zika virus, and probably only over the past few hundred years has spread out of Africa to the rest of the tropics. And so our native fauna, other animals, insects, haven't evolved with Aedes aegypti for very long, and they're probably not very dependent on them. So I suspect that if we eradicated Aedes aegypti, we would see little, if any, effect on other things. And there are other urban mosquitoes that are available to eat if you're a bat or a dragonfly or whatever. We have plenty of Culex mosquitoes in the U.S. That's why we have West Nile, for example, that would still be available in cities and urban environments. So, yes? Sure. So again, these are assumptions based on long experience with other sexually transmitted diseases. We assume that condoms, if used properly, will protect against Zika virus transmission, and that's what the CDC is recommending. Of course, abstinence is 100%. Condoms are 99 point something percent, but I think just condoms alone would be a big step. You know, the situation in Latin America is quite different than in this country with contraception and cultural views and so forth. But I think that if people either abstain or use condoms during that high-risk period, right now we don't know how long it lasts, so they probably should be doing that throughout the pregnancy, but that would eliminate virtually all of it, of the risk. Whether other contraception methods would have any effect is much harder to predict at this stage. We'll have to do animal experiments to determine that. Right. Okay.